lecture we discussed about post-fertilization events. In this lecture also we will continue with post-fertilization events only. Post-fertilization events, right? So, all the structures and events which occur after fertilization, they come under post-fertilization event. But the major events under post-fertilization are endosperm formation. What is endosperm? It is a nutritive tissue. The embryo should get nourishment from the endosperm. So, the first point under post-fertilization will be endosperm development. The second point will be embryo development. And the third point will be the ovule getting converted into seed and outside ovary getting converted into fruit. So, these are the major post-fertilization events. So, if we have to list down the major post-fertilization events means, what are they? What did I tell? There are 4 major post-fertilization events. The first one is developing the nutritive tissue, which is called endosperm development. So, I am writing the first point as endosperm development. The second point will be the zygote developing into embryo. So, that will write down it as embryo development. The third point will be the ovule getting converted into seed, means maturation of the ovule into seed. So, the third point will be maturation of the ovule into seed. And the last point will be maturation of the ovary into fruit. The last point will be maturation of ovary into fruit. These are the major post-fertilization events. Now, in this session, we will talk about the first one. What is that? Endosperm development. Take a screenshot and then we will take a fresh board and we will talk about endosperm formation. Right. So, our first point is endosperm development. Do you know what is endosperm? When do we use a word called endosperm? So, the first one is endosperm development. Right? Now, the endosperm is the name given to a food laden tissue, food depositing tissue, food storing tissue. Okay. So, what is endosperm? If I write E, that is endosperm. Endosperm is the name given to food laden tissue, or we can tell it as food deposited tissue. It is a name given to the food laden tissue which is meant to provide nourishment to the embryo, which is, what is its function, Amma? Which is meant, its role is to nourish. Whom it has to nourish? The embryo who is growing inside. So, its role is to develop the nourishing embryo. That is why embryo development, endosperm development precedes the embryo development. Means endosperm development will happen first. After that only embryo will develop, because when the embryo is developing, it needs food. Who will provide food? Endosperm is providing food. That means endosperm should already be there. So, that is why endosperm development precedes embryo development. Hence, endosperm development precedes embryo development. Embryo development. Children, this is a question in NCERT. They asked you a question like, why is endosperm development preceding embryo development? Means, why is endosperm development happening first and embryo development happening next? So we don't want the embryo to starve. So we want to provide food for the embryo. So that is why first we are doing endosperm development, then we are doing embryo development. Understood? Now we know that gymnosperms and angiosperms have dominated the plant kingdom. Now in gymnosperms, what do you mean by endosperm? In angiosperms, what do you mean by endosperm? So we'll use blue. In gymnosperms, the endosperm, it is a representation of female gametophyte. In gymnosperm, endosperm represents female gametophyte. Endosperm is nothing but, I am telling, it is represented by female gametophyte. Now all of us know that the female gametophyte means haploid, sporophyte means diploid. So female gametophyte means it is haploid. That means in gymnosperms, endosperm is haploid. So since endosperm is nothing but the female gametophyte, in gymnosperms we tell that the endosperm is haploid because it came from the female gametophyte and it is developed before fertilization itself. That means endosperm development or endosperm formation in gymnosperms is a pre-fertilization event. So in gymnosperms, endosperm development is a pre-fertilization event. Before fertilization itself, endosperm is formed in gymnosperms. Now let us talk in angiosperms. What is the scenario? In angiosperms, right, endosperm is formed. Come on, recollect, double fertilization. Remember about vegetative fertilization. Now we say that in this large central cell, two polar nuclei are there. These two polar nuclei will fuse with themselves to become a secondary nuclei. So these are polar nuclei. They are fusing to become a secondary nuclei. Children, polar nuclei is N and N. When you fuse, you will get a diploid tissue called 2N. Now, meanwhile, through the pollen tube, the male gamete is coming. One male gamete comes and fuses with the egg here. The other male gamete comes and fuses with this. So, if this male gamete also comes and fuses, 1, 2, 3. So, will it become triploid? Yes, it is 3N. And it is called as primary endospermic nucleus. What do you call it as? PEN. What do you call it as? PEN. Primary endospermic nucleus, which is triploid. Where is all this happening? In this large central cell. Now this large central cell, after the PEN is formed, converts into endosperm cell. It converts into endosperm cell, right? Now, like this, endosperm is formed in angiosperms, and this process of is called triple fusion. Triple fusion means 1, 2, 3 nuclei are fusing, and it is fertilization since the male gamete is involved. But which type of fertilization? It's vegetative fertilization. So, in angiosperms, endosperm is formed as a result of, in angiosperms, endosperm is formed as a result of vegetative fertilization. It is formed as a result of vegetative fertilization, which is also called triple fusion. Vegetative fertilization is also called triple fusion. Since 3 nuclei are fusing, hence, and in angiosperms, endosperm is triploid. Hence, the endosperm is triploid. The endosperm is, we'll write it here, it is triploid. Triploid means, children, 3N. It is triploid. And when is the endosperm getting formed in angiosperms? After fertilization, right? Because one male gamete came and fused with the egg. It resulted in formation of zygote. After zygote formation, pen is getting formed. Means in angiosperms, endosperm formation is a post-fertilization event. In gymnosperms, what did we tell? It is a pre-fertilization event. So, I am writing here, it is post-fertilization event in angiosperms. It is a post-fertilization event in angiosperms. It is triploid, the second difference, and it is formed. One difference is post-fertilization, the other difference is it is triploid, and the other one is it acts as a nutritive tissue. Understood? Take a screenshot fast. Right? Now, hope you understood what is endosperm. Now, let us see the types of endosperm. Depending upon the formation of endosperm, angiospermic endosperms are of 3 types. Let us see them also. Depending on the type of formation. Yes. Depending on the type of formation angiosperm endosperm is of 3 types. Angiosperm endosperm, I am telling it is of 3 types. Let us see what are the 3 types. First one is called nuclear endosperm or free nuclear endosperm. The second one is called as cellular endosperm. The third one is called as halobial endosperm. So, what are the 3 types of endosperm formations? What is the first one, children? Free nuclear endosperm. And then what is the second type then? The second type is called cellular endosperm. And what is the third type? The third type is called as halobial endosperm formation. Now, let us see what are these 3 types of endosperm. Among these 3 types, which is the most common type, means free nuclear endosperm formation is the most common type. Shall I write it here? It is the most common type of endosperm development. It is the most common type of endosperm development. So, how does it happen? Means, let me draw that embryo sac once. This is the embryo sac. This side is the micropylar end. This side is the callosal end. And antipodals, synergids, everything it got degenerated. Now, here we have the zygote. What is this called? This is called zygote. Zygote has formed at the micropylar end. Now, here we have endosperm, right? That triploid pen is there, no? Primary endospermic nucleus. So, what is this? This is pen, primary endospermic nucleus. And what is this cell called? This cell is called endospermic cell. The cell is called endospermic cell. Now, what happens? Since we are telling it is free nuclear endosperm, this primary endospermic nucleus, it undergoes mitotic divisions, but all the mitotic divisions are free nuclear divisions only. Only the nucleus divides, but the cytoplasm doesn't divide. As a result, you know what will happen? We will show the zygote and embryo in a separate color. And then what happens to this primary endospermic nuclei? Pen is doing only nuclear divisions and it makes many nuclei like this. It is making only nuclei and cytoplasm will be there, and here and there vacuoles are there. These are the vacuoles. So what it is doing here means it is undergoing, so these are mitotic divisions only. These are mitotic divisions only, but they are only nuclear divisions. They are only free nuclear divisions. So that is why the nucleus is keeping on dividing. Now after some time what happens means all these nuclei will join, all these vacuoles will join to make a large central vacuole. I will show it here. Now then what happens means the zygote is still like this at the micropylar end, and these nuclei will be pushed peripherally. Since the vacuole has, large central vacuole has developed, all these vacuole fuse children to make a large central vacuole. So this is a large central vacuole. The vacuole will occupy the space, then the nuclei are pushed to peripheral, and cytoplasm is also there. Cytoplasm means in between this cytoplasm is there. Now this cytoplasm grows later on. What happens? The nuclear divisions are continuing. That's what you can see. Here one nuclei became 5 nuclei, here they became many nuclei. Later on the cytoplasm thickens. After this, what happens? So this is the large central vacuole formation. Now after the large central vacuole is formed, cytoplasm thickens. The cytoplasm thickens. When the cytoplasm thickens, then it undergoes cytokinesis also. It undergoes means cytoplasm, when the cytoplasm thickens, vacuole will disappear. Vacuole disappears. When the vacuole disappears, the resultant structure what we see is, meanwhile the zygote also starts developing, and the zygote has a suspensor cell here, it has haustorial cells here, and it has a pro-embryo here. So what is this? Now this is the embryo. Zygote has developed into embryo. Now what happens here? First vacuole developed and pushed the nuclei towards the periphery. Now then the cytoplasm comes and occupies the space and it gets thickened, and then meanwhile these peripheral nuclei undergo cytokinesis. They make their walls. They start making their walls. So then the peripheral nuclei, as they start making walls, it becomes cellular endosperm, right? So see, I am making, so this is one cell. Now in each cell start keeping one nuclei. So the peripheral nuclei are surrounded by walls now, means the free nuclear division, now it became cellular division. Cytokinesis happened. Now can you see the outer cells, outer nuclei are surrounded by walls. Slowly, initially the inner ones, free nuclei only will be there. So just I am making the outer ones are cellular, whereas the inner one still nuclei will be there. Free nuclei will be there. Okay. So now this is what, when we drink a tender coconut, in the tender coconut, the water what we drink, so what is this part called? Coconut water. The coconut water which is inside represents, what does it represent? It represents free nuclear endosperm. Free nuclei are there. So in tender coconut, the coconut water represents free nuclear endosperm. It's liquid, right? So it can also be called as liquid endosperm. It is also called liquid endosperm. As the coconut starts maturing, then the peripheral ones becomes white kernel, right? So what is this one called? This one is called white kernel, the white coat, right? The white kernel, it is liquid endosperm or solid endosperm? It is solid. And it is free nucleara, it is cellular endosperm. It is cellular endosperm. So white kernel represents solid endosperm, or we can tell it is cellular endosperm. It becomes cellular endosperm. And if you take a completely dried coconut, then even these nuclei also will be covered by cells. If we take a complete matured one, then all these are also covered by cells. Everything becomes cellular, right? Everything becomes cellular. So this is one type of endosperm development. It is called free nuclear endosperm development, because the initial endosperm divisions are only nuclear divisions. Karyokinesis, nucleus division is only happening. Cytokinesis happens later on. So that is called free nuclear endosperm formation, and it is the most common type of endosperm development. Where do we find it? We find it in rice, we find it in wheat. We wrote there, we can find it in coconut also, and we find it in sunflower, and we find it in a plant called Capsella bursa pastoris. We find it in Capsella bursa pastoris also. This liquid initially which is there is called liquid endosperm, and this liquid endosperm, what is it? It's free nuclear endosperm. We tell that that is a very good fluid. It is rich in nutrients. So it is rich in vitamins, it is rich in minerals, it is rich in sugars, rich in proteins, and this fluid is isotonic to our body fluids. The fluid is isotonic to our body fluids. So it is good for our body. Whereas this white kernel represents the solid endosperm. Understood, children? This is about the first type of endosperm development. Where do we find it? Rice, wheat, coconut, sunflower, and Capsella bursa pastoris. Take a screenshot, then we will talk about the second type of endosperm. Right now, can I rub this? Can we see the second type of endosperm development, which is called cellular endosperm development? In free nuclear endosperm development, the first divisions are nuclear divisions only. 1 nuclei became 2 nuclei, 4 nuclei, 8 nuclei. Later on, after very long time, they became cellular endosperms. But here, in cellular endosperm formation, the first division itself is cellular, means karyokinesis, cytokinesis proceed simultaneously. So, what is cellular endosperm? Here we are telling that the primary endospermic nucleus, which is triploid. So, it undergoes simultaneously karyokinesis and cytokinesis. Pen undergoes simultaneously both karyokinesis means nuclear division, cytokinesis means cytoplasm division. Simultaneously both karyokinesis and cytokinesis. It is undergoing both karyokinesis and cytokinesis, means nuclear division, cellular division also. Then it will completely become a cellular endosperm itself. If we have to see the picture, again let us draw the embryo sac. Micropylar end, callosal end. This is the zygote. This is zygote. 1, 2, 3, triploid pen. It is. Now this pen, it starts dividing and it makes 2 cells, 4 cells like that. So when we have to draw 1 cell, another cell, another cell, another cell, right? Here is the zygote. This is zygote again. So can you see? They are not free nuclei. Nucleus within the cell. Means this type of endosperm development is called cellular endosperm. this type of endosperm development. What is it called? It is called as cellular endosperm. In which type of plants do we find cellular endosperm? Examples are, one is balsam, next one is datura and petunia. In balsam, datura and petunia, cellular type of endosperm development will be there. Means it's not like once it will do only nuclear divisions, later it will do cellular divisions. Here it will simultaneously do nuclear division, cytoplasm division, nuclear division, cytoplasm division. So the cells are increasing in number. So it is called cellular endosperm. The example is balsam, datura and petunia. Take a screenshot, then we'll talk about halobial. Right? Now can I erase this? Now what is halobial? So this makes some sense, like free nuclear means we can make out. It is undergoing only nuclear divisions. Cellular also we can make out. It is doing cellular divisions, mitotic divisions. Then what is halobial endosperm? How did it get the name called halobial? In monocots, there is an order called halobiales. So those plants which belong to the order halobiales exhibit this type of endosperm formation. That's why it is called halobial endosperm formation. So children, where is it seen? It is seen in order. What is that order I told? Halobiales. It is seen in the order halobiales of monocots. So that is why it is called halobial endosperm formation. Now we got a clue what we meant by halobial word. Now which type of endosperm formation it is? Is it free nuclear or cellular? Means it is somewhere intermediate between this to this. So we consider it as intermediate between nuclear and cellular type. It is intermediate between nuclear and cellular type. We will draw the diagram. Through the diagram, we will try to understand. So here what happens is, this is the zygote, pen, micropylar end, callosal end. Here what happens is, the endosperm cell, this is endosperm cell. The endosperm cell undergoes a transverse division. The endosperm cell undergoes a transverse division, means like this. It undergoes a transverse division, yeah, but it is not an equal division, ma. It is an unequal division. One small cell and one big cell. It is an unequal division. The micropylar end endosperm cell is the bigger cell. The callosal end endosperm cell is a smaller cell. What is this? This is micropylar end and it is the large endosperm cell. And what is this? This is callosal end small endosperm cell. Okay. Now, next what happens means they undergo divisions, right? This is the zygote. Now, this large endosperm cell which is towards a micropylar end is functional. This is functional. The large endosperm cell is functional and this cell is vestigial. It degenerates. This endosperm cell which is towards the callosal end will degenerate and the large endosperm cell which is towards a micropylar end will develop. It is developing and initially it shows free nuclear divisions. Then if it is showing free nuclear divisions, then these are the nuclei. You remember when will the nuclei push towards the periphery? When the central space is occupied by a large central vacuole. When the central space is occupied by a large central vacuole, then they will be pushed aside. Afterwards, what happens? This grows into embryo. These are the suspensor cells. And these are the embryo cells. And then it becomes cellular endosperm. The vacuole disappears and it is making a cellular endosperm. Now, all this becomes endosperm tissue. all this becomes an endosperm tissue. Now the embryo, see the embryo is inside and it is taking nourishment from this endosperm tissue. Children, such a type of endosperm formation is called as halobial endosperm, and it is seen in an order called halobiales, which comes under monocotyledonate class, and the endosperm development is intermediate between the nuclear and the cellular thing. If we have to write an example, the example is asphodilus. If you have to take an example of a plant for halobiales, it is asphodilus is an example. Take a screenshot, we'll continue. Right? So we understood the types of endosperm. Now let us talk about the fate of the endosperm now. With examples, we have seen all the three types and we came to know the common type also. The common type is free nuclear endosperm. We have seen the example of coconut water and that coconut white kernel. Coconut water is liquid endosperm. The white solid what we eat is cellular endosperm. Now, what is the fate of the endosperm? What is the fate of endosperm? Now, the endosperm's outside portion will be nucellus. As the endosperm is growing, the endosperm will crush off the nucellus and endosperm will occupy. And this endosperm will be there, or this inside the endosperm, I am drawing embryo and the embryo will eat it off. Now, endosperm is crushing off the nucellus and this in turn this endosperm is eaten off by the embryo. So, as endosperm development continues, nucellus gets crushed. It will not be there. Nucellus gets crushed. And this endosperm, inside the endosperm, who is there? We are drawing embryo is there. And this embryo starts consuming this endosperm also. Okay. Nucleus gets crushed and the developing embryo consumes the endosperm. The developing embryo needs food, endosperm has reserve food, it starts consuming the endosperm, it will be consumed off. Now, there are two types of seeds we know, monocot and dicot seeds. So, one is the dicot seed and the other one is the monocot seed. You know, the dicot embryo, during its development, it will completely consume the endosperm. It will not leave little endosperm also. So, that is why dicot seeds are called as non-endospermic. Dicot seed has eaten off the endosperm. So, that is why dicot seeds are non-endospermic because they consume the endosperm. So, we have another synonym. It is also called albuminous. No albumin, no protein content means what is there inside the endosperm? Protein is there. Albumin now it is not there because it is consumed off. Dicot seeds are called non-endospermic seeds or albuminous seeds. Whereas come to monocot seed. Monocot seed, it will not consume the complete endosperm. It will retain endosperm. Little more bulky endosperm it will retain. That means only little endosperm it will consume. That means monocot seeds are endospermic then. So, monocot seeds are, what should I write? They are endospermic. Endospermic means albumin is there. So, write them as albuminous seeds. They are also called albuminous seeds. Now, list down some examples of dicot seeds. Pea is an example. Gram is an example. Groundnut is an example for dicot seeds which don't have endosperm. List down some examples of monocots. Rice is an example. Wheat is an example, right? Then rice, wheat, maize, all these things are examples of monocots. Now, But science without exceptions is not possible. Means now I am going to tell a dicot which has endosperm. Actually, dicot should not have endosperms, but castor. What is castor? Castor is dicot. Castor is dicot, but has endosperm. But it has endosperm. Whereas we have to list monocot, which is non-endospermic. So, orchidema. Orchids. What are they? They are monocots. Orchids are monocots without endosperm. They are monocots without endosperm. So, these are the exceptions here and exception here. With this, we covered everything related to the first point of post-fertilization events, which is endosperm development. Then what is our second point? Our second point is embryo development. So, post me in the comment box how many types of embryos are there. We will discuss about the embryo development in the next session. If you like the content, like, share and subscribe to my channel. Thank you.